Well, hello, ladies and gents. I hope you're doing well today. So, what I'd like to do is run through concepts that I know will appear on the test. I sat down with the multiple choice and true false part of the test, as well as the short answer part of the test, and I made a list of concepts that appear. My original plan was to write on the screen and use this pen I purchased over the internet, but it's lousy. It does a lousy job. So instead, what I did was I just hand wrote things and snapped some pictures and want to discuss those with you. So I hope that will suffice for review for you. So the first thing I want to do is make sure that we highlight differences between anatomy and physiology. Anatomy focuses on the structure of living materials, and physiology focuses on sort of the chemical reactions or the processes uh, that we associate with life. So I think the shorthand we often use is anatomy is structure, physiology is function. So if you're looking at a preserved specimen, if you're doing dissection, if you're looking at prepared slides, if you're looking at a model like the model we had with the body cavities, that's all focusing on anatomy, how things are built, where things are located. Physiology focuses on what things do. Enzymes can catalyze chemical reactions, gases can be exchanged. Um, you can have hydrolysis, dehydration, synthesis, that kind of stuff. So anything related to structure is anatomy. Anything related to what things do or function is physiology. So make sure you're aware of that difference and that you can cite examples of each. Next, we're going to look at types of bonds. Now, ionic bonds result from the transfer of electrons between partner atoms, or in this case, ions. So in the example we have there, the sodium ions, or a sodium ion, will give up, uh, sorry for this, I always have great plans, but then the plans may not pan out. Okay, call this good. So the sodium donates an electron to chlorine, and that creates these ions: a positively charged sodium ion because it lost an electron, and a negatively charged chloride ion. And then, since opposite charges attract, it's that attraction that holds together those ions. That can work great in an environment that doesn't have a lot of water, but in the watery cell, water, since it's polar, can break or dissociate those ions and dissolve them. Now, next we'll look at polar covalent bonds. In the case of polar covalent bonds, electrons are being shared, right? But the electrons are shared unequally. So in the diagram we look at with that, the electrons spend more time around partner 2, less time around partner 1. So because the electrons are shared unequally, then you wind up with a partial negative charge on the partner that is more electronegative or has more of those electrons, and a partial positive charge on the neighboring, or the other portion of the molecule that has less time with the electrons. Now, contrast that with nonpolar covalent bonds. In the case of nonpolar covalent bonds, the electrons are being shared equally. And if they're being shared equally, then there is no unequal distribution of charge throughout the molecule. Now, polar covalent bonds can result in, well, let's see what happens here. We may just have to live with this. Okay. Polar covalent bonds result in hydrogen bonds between neighboring molecules. So again, a hydrogen bond is not technically a bond. It's called an intermolecular force. It's an attraction between neighboring molecules. And what happens is, in this case, the partially negatively charged oxygen will have an attraction to the partially positively charged hydrogen in a neighboring water molecule. And what that does is create some of the properties we associate with water. So it makes water behave in sort of peculiar ways. All right. Next, let's take a look at the body systems. You guys had that systems poster, and then you did the systems poster comparison essay. It's going to be important to know what all the systems do and how they interact with one another and what happens if one breaks down. So that's basically what we did in that essay. So if we look at the urinary system, it runs through your kidneys and it removes nitrogenous waste from your blood. So your blood has toxins from your cells, and then your kidneys filter out those toxins, including a bunch of nitrogenous waste, and that stuff exits the body in the urine. Your digestive system breaks down your food. Remember, you're made of what you've eaten, right? So your digestive system breaks those molecules down into smaller pieces or monomers through hydrolysis reactions. Your endocrine system uses hormones to communicate between cells of the body. So you can send out these chemicals, and then cells with receptors for those hormones can respond in certain ways, usually to help you maintain homeostasis. Your muscular system, muscle attached to bone, and muscle contracts, so it just moves you around or moves things through you. Your respiratory system has one function, swapping out gases. You release CO2 and you take in oxygen. Your nervous system is a fast acting communication system. What it does is detect stimuli, makes a decision about how to respond, and then sends those instructions out through the afferent pathway to tissues of the body to respond. Your circulatory system transports materials through the body. You're made of about 10 trillion cells. And so to get waste out of there and raw materials in fast enough, you need a circulatory system that runs really closely to cells. So your circulatory system transports that material really quickly through your body. Reproductive system, it's for making babies, so you make a new generation. Lymphatic system, your circulatory system leaks fluid all the time. And so your lymphatic system is like the sewer system that collects those fluids back and then returns them to the circulatory system. Your integumentary system, skin and glands associated with that, so it helps protect your body. It also helps with like regulating ion balance and temperature. Your immune system has a protective function as well. It will protect you from pathogens that have entered the body. And your skeletal system, it sort of lends support to the body. It's something to which organs can attach to, and also is protective. Think of like your rib cage protecting your heart and lungs, or your skull protecting your eyes and your brain. All right. So that's first set of ideas. Let's move on to this second document here. Features we associate with life. To be alive, you need to have a metabolism. That means you need to take in raw materials and get rid of waste. So basically, you've got concentrated chemical reactions going on inside of cells. Reproduction is a feature of life. Now, it's not just about making new organisms. It's about making copies of cells in your body. So you're constantly turning over cells. And so reproduction of cells is one feature of that. Let's see. All organisms are made of cells, so you're made of this non-living stuff, carbs, lipids, proteins, nucleic acids. When you put them together, you got these cells that are alive. There is organization, so you can take things like amino acids from the environment and then turn them into something more organized like proteins. And then growth and development occurs. When you started your existence, you start off as a single cell, and now look at you, trillions of specialized cells. All right, next thing we'll look at is concept 5 here. That's related to feedback loops. Okay, so there is the stimulus. The stimulus can happen inside your body or outside your body. And what it can do is pull your body away from homeostasis. Now, receptors throughout the body detect that. You have receptors for touch, pain, temperature, pH, whatever the case may be. And those receptors detect what's going on in and around the body. And then it sends a signal through the afferent pathway. Remember, the afferent pathway goes at the central nervous system. And then the control center would be your brain and spinal cord where decisions get made. Once an appropriate response is determined, then instructions are sent out through the efferent pathway. And then the efferent pathway carries electrical signals to an effector, which is basically a muscle or gland that does something, allows you to respond. Now, how does the body respond? That's the topic in number 6 here. The body responds by either bringing you back to homeostasis or pushing you further away from homeostasis. So homeostasis is a steady state. Okay, so steady temperature, steady oxygen saturation, steady pH, whatever the case may be. And what will happen with negative feedback is if your body moves away from homeostasis, negative feedback brings you back to homeostasis. So if you're trying to solve problems on the test, if it brings you back to homeostasis, that's negative feedback. Positive feedback, on the other hand, continues to push you away from homeostasis. So whatever is produced will actually enhance its own production. Okay. Let's move on to number 7. Hey, know your directional terms. Okay. So you guys have done some stuff related to that. Let's see if I'll push my luck. See if I can zoom in anymore. Okay. So Terms to remember: superior means toward the head, inferior means toward the feet, medial means toward the midline, and lateral means away from the midline. Let's see. Posterior means toward the back, anterior means toward the front. And then there are terms like deep and superficial. Deep means deeper inside the body, so that's our creepy looking smiley face there. So deep means away from the surface of the body, and superficial means toward the surface of the body. So if you have a superficial cut, it's real close to the surface of the skin. If you have a deep cut, well, it cuts in the deeper tissues. Another thing to know is the planes of dissection, the little XYZ planes. Sagittal, I should turn facing the camera more directly. Sagittal splits you into right and left pieces. And again, it's the organisms right and left. So just think sagittal splits you. Transverse cuts you into top and bottom pieces. So a transverse cut cuts you into top and bottom. And then frontal cuts you into front and back pieces. Okay, so definitely know your planes of dissection. Let's see. Along with other anatomical terms. Goodness. Okay, sorry guys. You've got these regional terms as well. It's really not liking that. Okay, let's call that good enough. So certain regions of the body. Patellar. The patella is your kneecap. So the patella is the front of the knee. Popliteal is referring to the back of the knee. Think about, again, popping somebody in the popliteal region, but don't do it actually. The axilla is the armpit, so the axillary region is referring to the armpit. Lumbar is the lower back, so good place to get your spring break tattoo or whatever. Antecubital is the front of the elbow, so that's where blood draws can be taken, is the antecubital region. Okay, we also talked about body cavities. Those are spaces inside the body where organs and tissues can be found. The two main subdivisions are dorsal and ventral. The dorsal cavity is basically the cranial and spinal cavities that hold your brain and spinal cord respectively. The front cavity, or the ventral or belly side cavity, is subdivided into thoracic cavity, which has the heart and lungs, the abdominal cavity, which has a lot of gut organs, and then the pelvic cavity, which has things like the bladder, the rectum, the uterus, things of that nature. All right. Idea 10, it's related to the pH scale. So the thing to remember, 7 is the magic number, right? 7 is the magic number because when you take water and split it apart, it creates a hydrogen ion, an H+, and a hydroxide ion, an OH-. So at 7, your hydroxide and hydrogen ion concentrations are equal, which means they can just reform and create water. So water will break apart and reform all the time, or not all the time, but it happens. So at pH 7, you have equal concentrations of those ions. Well, now what happens if you start to have extra hydrogen ions? If you have extra hydrogen ions, then the pH drops and it's in the acidic range. So again, pH below 7 means it's an acid, which means it has extra hydrogen ions. Now, the further you get from 7, or the farther you get from 7, the more and more hydrogen ions you have. So as you approach 0, you have a really high concentration of hydrogen ions. On the flip side, if you go above 7, or have a pH above 7, that's the alkaline or basic end of the scale. And the closer you get to 14, the more OH minuses you have. Now, the issue with this, having a lot of hydrogen ions or a lot of hydroxide ions, a lot of H+ or a lot of OH-, is that those ions will break bonds. And if you have more ions, you're going to break more bonds. If you break more bonds, you're going to destroy tissue. So strong acids and strong bases are very destructive. Another thing you're going to have to do on the test is calculate the difference in ion concentration as you move to different pHs. So let's say you have one substance with pH 2 and the other with pH 7. Well, again, easy. You could do like 10 to the 5th. But I would say the easiest thing to do, and the notation that'll be on the test, is like what you see in this example here. So I found the easiest thing you can do is write down the range, 2, 3, 4, 5, 6, 7, and then know that every step is a factor of 10. So you're basically adding a 0. So if you go from a pH 2 to a pH 7, it's 10, 100, 1,000, 10,000, 100,000 times as many hydrogen ions. So again, imagine all the bonds that can be broken and the destruction that can be wrought on tissues of the body. All righty. Last page here. unless you want to just go through it all in reverse. Let's see. It's the hydrolysis and dehydration synthesis reactions. Okay. Remember, dehydration synthesis, to dehydrate is to remove water, and to synthesize means to create. So you're forming bonds. Oh, shoot. I reversed breakdown and build up. Please scratch that and know that I messed that up when I was writing today. So with the dehydration synthesis reaction, you notice what happens. You have the shorter polypeptide there. It's got two, or not, I should say polypeptide, the shorter polymer. It has two molecules with an OH, and then the monomer just has a little H there. Well, you remove the H and OH, and that forms water. And in the process of doing that, you create a bond. So again, please ignore where it says breakdown and dehydration synthesis. It should be built up. So with a dehydration synthesis reaction, you remove water, form bonds, and you make more complex substances, more complex molecules. With hydrolysis, hydro means water and lys means to split or to break. With a hydrolysis, think you break with water. You add water, and when you do that, it breaks the bonds. So again, you look at the polymer, you can break that bond, and you keep the monomer separated from the polymer by adding the H and OH. Okay, so dehydration synthesis, you remove water. Hydrolysis, you break with water. And when you do that, you can create monomers and polymers. So there you see the polymer made of many monomers linked together. Through hydrolysis reactions, you break the bonds and you create these monomers. And then through dehydration synthesis, you put the monomers back together and create new polymers. Let's see. Speaking of polymers, let's look at the macromolecules. Okay, looking at carbs. Carbs are made of CHO. The monomer that we'll pay attention to is glucose. And the relevant polymers that we'll look at are starch and glycogen. Starch is how plants store extra sugar. Glycogen is how we store extra sugar. And again, when we're thinking of functions of carbs, focus on the energy. Lipids, they're also made of CHO. They technically don't have monomers or polymers, so we'll just focus on function. For function, the way I remember lipids is I think membranes, energy, and messengers. So the membrane is a phospholipid. The energy is triglycerides. Those are like saturated, unsaturated fats. And then the messenger is hormones. Proteins, they're made of chons. They have nitrogen and sulfur. Amino acids link together to form these big proteins. And they can do all sorts of things. They can be enzymes that speed up chemical reactions or catalyze chemical reactions. They could be antibodies. It could be collagen. It could be pumps and channels and cell membranes. Like proteins do a lot of things. Finally, nucleic acids. Nucleic acids are made of CHOMP. They have nitrogen and phosphorus. The monomer is a nucleotide. Nucleotides are like ATCG. You're adding guanine, thymine, cytosine. And then the polymers are the DNA and RNA molecules that we're familiar with. Now, what do? DNA is instructions for making proteins, and RNA actually does the job of making the proteins. All right? Now, again, this is not necessarily an exhaustive list of everything, but just going through the test today, like these are the things that stood out to me. And again, I tried to make something sort of visual with this. So I hope this helps with your review. If you happen to have any questions, please let me know. Otherwise, have a great day and enjoy your weekend.